Okay. David, Keefe. Professor Keefe, how do I introduce you? You're a big star. You're a big star. I'm going to take 10 minutes to introduce you.
No, please.
Department chair of NYU, and of course, so apt in all aspects of assisted reproduction. It really is a pleasure. We should have you at every meeting. At every meeting, you should be in the front row, so you can ask your excellent questions.
Great.
David.
Thank you. Thank you, Jacques. It's so refreshing. Jacques Cohen is always ahead of the curve. It's so fascinating to hear where he's going. I tell you, that it totally relates. I am the chairman of the department, so we have 155 faculty, and we have something called Enterprise Connect, which is similar to what he's proposing for the lab, and I get an alert if one of my faculty's profit loss margin drops below a critical level. Seriously. And then that's what I was doing yesterday on the phone. It's like an emergency. And isn't it ironic that we don't do that for something like IVF, which is essentially an intensive care unit for embryos, right? So it's brilliant. Absolutely amazing. So, all right, we talked yesterday about genomic instability, how ubiquitous it is, aneuploidy mosaicism, partial mosaicism, copy number variants. It's everywhere in the preimplantation embryo. So what are the contributors to this global genomic instability that we see in preimplantation embryos? We've seen a lot of discussions about spindles, chromosomes, cytoplasm, and I'm going to kind of run through this, and it's been said that basic scientists have all the answers, but clinicians have a few good questions, and I'm a clinician, so what I'm going to put out there are essentially questions, not definitive answers, but questions. Our group was actually the first one to report a deletion in mitochondrial DNA in human oocytes in older women back in 1995. In fact, that was my dissertation for my endocrine boards. But after doing a number of different experiments, when Lynn Liu and I started working together, we did what we thought was an important experiment. I was taught by Nina Schwartz, who was a famous reproductive biologist back in the 60s, 70s, to do the experiment that's going to disprove yourself first. So hoping that nuclear transfer would rescue the reproductive aging phenotype, that we would in fact be able to demonstrate our hopes that the mitochondrial DNA would be rescued when taken from a young oocyte into the old. We did nuclear transfer, reciprocal nuclear transfer in an aging mouse model, and here you can see in the young, the white bar is essentially fertilization rate, and then the gray and black bars are what looks like aneuploidy in a mouse. We have spindled abnormalities and congression abnormalities with the chromosomes. So the young, high fertilization rate, low aneuploidy rate, and the old, a very high fertilization rate and a very high aneuploidy rate in this. This was a senescence accelerated mouse. So look what happened when we used young cytoplasm, that's YC, in old nucleus. No difference. We did not see any rescue. This was devastating to a young assistant professor at Brown. But in the laboratory I used to occupy was formerly occupied by Dr. St. Georgi, who won the Nobel Prize in 1971. And it says study, there's a big graph, a big chart right over the Xerox. It says study nature, not books. So we then went on to figure out why that might be the case. The first thing, there's almost no oxygen consumption early on. There's up until the blastocyst stage, the black dots are oxygen consumption measured by the non-invasive oxygen probe. It's now called Fertilase. People, I think, have attempted to use it clinically. But here we found there was almost no oxygen consumption, which is the sine qua non of oxidative phosphorylation. And in fact, multiple studies have shown that oxidative phosphorylation, what the mitochondria does, is not actually the principal source of energy. It's actually, there's a number of different pathways. Adenosine salvage pathway has recently been proposed, been demonstrated. It's thought that the oocyte and the early embryo are in a different mode. They're not in an energy-producing mode. In fact, the mitochondrial DNA is too precious a payload to contaminate with reactive oxygen, which is the inevitable byproduct of oxidative phosphorylation. It uses the pentose phosphate shunt pathway. It uses pathways that are involved in synthesis. It has so much synthesis to do that there's almost no oxidative phosphorylation. And in fact, you can experimentally reduce the number of mitochondrial copy numbers down to incredibly low levels, and it has no impact whatsoever on blast development. Now, interestingly, a lot of folks, John van Berkel and others, actually cite this paper as demonstrating the critical role of mitochondria in development. It's true. Post-implantation, there was a massive impact of reducing mitochondrial DNA copy number. By the way, the way they do that is they make the mouse haploinsufficient for the gamma polymerase, which is the polymerase that replicates mitochondrial DNA. But you can drive the copy number down to 10-fold below, and you get normal blast development and no aneuploidy. That's mouse. It's true, and it's in fact inbred mouse strains. But what about humans? There's actually a natural experiment. God or Darwin, whoever makes mitochondrial DNA mutations, did the experiment. There are babies that die because they're nearly homoplasmic for mutations that essentially knock out oxidative phosphorylation. But their mothers are perfectly fertile. This is data that comes from, it wasn't highlighted this way, but it's embedded in a report from the group in Newcastle that's proposing pro-nuclear transfer. We're going to hear more about this in the next session, but essentially they showed that they have the largest sample in the world of pedigrees of mitochondrial DNA mutations, mother-offspring transmission, and there was no difference in the fertility rate, the live birth rate, in those pedigrees with the harboring deadly, detrimental, toxic mutations compared to age-matched controls. No difference, no impact. And so that's why we moved to the nucleus. Not that I didn't understand mitochondria, not that I didn't hope and wish and dream someday of a treatment. And again, doesn't mean it's over. It's just a question. Because there's a lot of stuff in cytoplasm. Is it cell cycle? Is it calcium waves? Is it the mitochondria involved in calcium uptake? I just don't think it's oxphos. So I think it's a really important question to answer. If it works, then why would it work? To me, the prediction is it won't. So we've moved to where the money is. Why do you rob banks? Because there's money there. Why would you look at chromosomes? Because that's where chromosome non-disjunction mosaicism originates. So we focused on the telomeres because they're a major source of non-disjunction, of mosaicism, of all copy number variants. Is an enormous literature mainly in the cancer field, basic biology field. What are telomeres? You probably all you folks that are younger than us older folks already knew about this, but it had actually been predicted by Watson when he discovered the structure that there's a problem at the end of a chromosome because the chromosome ends, the DNA ends in a blunt end, and that's like a double-strand DNA break. You can't distinguish between the end of a chromosome and a double-strand DNA break, and it would trigger, and it does trigger, the DNA damage response. So nature has evolved from plants to humans a conserved mechanism to protect the chromosome ends. It also has another problem when you replicate DNA. It uses in the lagging strand synthesis the so-called Okasaka fragment, RNA primer that needs to be lifted off before the leading strand can be used to elongate the end. And what happens is the ends of a chromosome, to solve these two issues, they form a loop. It looks just like, by the way, a bacterial viral DNA. It's a loop. And in fact, that loop that doesn't just sort of curl like a curly hair. It actually is regulated. There's a number of proteins called, they form a shelterin. It's a protein DNA matrix that essentially forms that telomere loop, and protects the end, blocks the DNA damage response. And you can experimentally uncap the telomere. You can remove that shelterin one by one. And when you do that, you activate ATM signaling, ATR signaling. You trigger something called non-homologous end joining, which ends up causing these long loop. So look what happens when, this is from Aniel Safir, who's a colleague at NYU Langone. She experimentally in human cells, these are cultured cells, not embryos, but she experimentally pulled off one by one each of the shelterin components until, look what happened. These are each individually chromosomes, and they've completely stuck together like sausages. Now, obviously, we don't see that in humans, but this is just a proof of principle that effectively, just by merely removing these proteins one by one, which is effectively removing the telomere loop, you get complete chromosome fusion. Does that happen in humans? Probably not in embryos to that extent. Does it happen in one chromosome? Does that result in mosaicism or partial mosaicism? We're not sure. We know that this is a problem, that telomeres are a problem during early development. By the way, not in the male genome. In the male germline, Telomerase, the enzyme that elongates telomeres, the reverse transcriptase that uses an RNA primer with a reverse transcriptase catalytic component, is capable of fully reconstituting telomeres in the male germline throughout the life of the male. It's immortal. In fact, interestingly, male germline telomeres actually elongate. They don't shorten with age. They don't even stay the same with age. They elongate, which is one of the great injustices in the world. But men also remain fertile. We know that there's subtle decreases in motility, in morphology, and concentration, but male fertility remains, and we all have heard the stories of the Charlie Chaplins of the world that have children in their 70s and 80s. But in the female germline, this is in a way a terminally differentiated cell type. It's not a stem cell. The primordial germ cells were, and this is the group that originally discovered telomerase activity in humans, Woody Wright and Jerry Shea, and they looked, there's all sorts of different tissues, and this is just when they looked at oocytes, eggs, zygotes, all up to blast. In the early germline of the female, there's no telomerase activity or no appreciable telomerase activity, so the female of the mammal is sort of stuck with whatever telomeres it was endowed. And recall, it doesn't divide, so it doesn't go through reproductive senescence after entering meiosis, but it did divide. And in fact, when the late Bob Edwards heard me first talk about this, he came up and said, aha, the production line. I've been trying to figure out for decades why late exiting oocytes from the proliferative phase of oogenesis would end up having fewer chiasma and having more aneuploidy. And so we tested this. This is, again, with Lou. We looked at myocytes. We looked at chiasma and synapses using immunocytochemistry of their gonads in utero. And you can see it when we had telomere shortening induced genetically through the telomerase null mutation. You can see a tenfold difference in the synapses and chiasma. It's been known from yeast to plants to mouse to humans that telomeres are absolutely essential for formation of chiasma and synapses because of the important role they play in tethering the chromosomes to approximate like chromosomes of like size, and hence they form the initial homolog pairs, which then go through synapsis and then chiasma. When we shorten experimentally the telomeres in mice, which, by the way, they're very poor model. The average mouse is a very bad model for oocyte aging. They just don't have much of an effect. You get like 3% aneuploidy instead of the sort of up to 60-70% you see in humans. But when you shorten the telomeres, genetically or pharmacologically, you get massive amounts of fragmentation. You get arrest. You get very low blast rate, very similar, but sort of phenocopies the human. And interestingly, so are the spindles disrupted. Now that at first pass makes no sense. We were all sort of assuming that spindles are cytoplasmic constituents. They come out of the tubulin that's soluble in the cytoplasm and then assemble. But actually, it turns out that spindles are stabilized by structures both in the kinetochore and the telomeres. The telomeres actually have all the components of the centromere, which forms the kinetochore. You can find all the components of the spindle assembly checkpoint, presumably because both centromeres and telomeres have a very similar evolutionary origin. So anyway, when we shorten telomeres, we found these are normal meiotic spindle in the mouse. And when we shortened the telomeres, it was a vast disruption of both the morphology of the spindle and also congression. Interestingly, in kind of a reversal of fortune, the fission yeast community has discovered that telomere disruption makes abnormal spindles in yeast, in the fission yeast. So over the last 5 years, they've sort of discovered what we discovered, and almost by chance we discovered this, and they're now dissecting the whole mechanism by which telomeres assemble microtubules into meiotic spindles. We also found extensive chromosome disruptions, including fusion. So we believe that the mouse is an interesting model in that it's almost like a tabula rasa. It really doesn't have much of an oocyte-based infertility, presumably because at least inbred mice have been bred for thousands of years. At first, for the sort of the royal interest of the Chinese Ming dynasties, and then later, for laboratory reasons. They have very poor oocyte infertility as a model. But by experimentally shortening the telomeres, we can phenocopy the reproductive aging in mice. It gives an explanation of how an assembly line or a production line might work. We can mimic the decreased chiasma you see in oocytes from older women. This is one of the ways by which you could generate or imagine having spindle abnormalities. And it shows how even low levels of mitochondrial DNA deletions, which uncouple low levels of ROS, might chronically over time contribute. We can essentially phenocopy the apoptosis and fragmentation. So what about women? That's mouse. Interesting. It's a model. It's a mainly genetic model. Well, recently we had the opportunity to study telomeropathy in women. Now, telomeres are so central to human health that the dyskeratosis congenita, which is a syndrome involving telomere dysfunction, is very, very, very bad for your health. It's lethal when it's homozygous. When it's hetero, it causes a severe syndrome. Any rapidly proliferating tissue, especially bone marrow, goes through failure. You get a lot of different various phenotypes. But this woman was a 30-year-old who was noted to have a precocious aging syndrome. She's actually a beautiful woman. She's from Sweden, but she looks like a beautiful 55-year-old. It was presented with aplastic anemia, was discovered to have leukocyte telomeres in the first percentile. So she had DKC. So she was referred to NIH in the rare disease unit where they're doing whole genome sequencing to identify the causes of these bizarre, unusual syndromes. And she expressed she had just gotten married and had a great interest in embryo freezing. So she was referred to us because they knew we were interested in telomeres and reproduction. And look at her AMH for the clinicians in the group. That's very low for a 30-year-old, as is her antral follicle count. Regardless, after talking to her hematologist, her cardiologist, all the different ologists, they agreed that it would be acceptable to give her a trial. So we ended up giving her the maximal dose. And by the way, she's très petite. She's like 5'1" and about 95 pounds. But still, we used 600 international units a day with an E2 prime mixed protocol. It took 18 days before we finally got 8 oocytes. We just blasted her ovary and finally got 8 oocytes and 2 blasts. One actually happened to be euploid. So we've got that. Interestingly, when we measured the telomere length in her the embryos that had arrested, we ended up seeing, markedly short telomeres, so that begs the question: how would you even have a normal euploid embryo? Well, this one clinical case actually brought us back to a paper that Lynn and I did a number of years ago, which is just essentially we started just profiling telomere lengthening in early development, and this was incredibly surprising to a lot of us. Look what happens in early development. So this is mouse in which we fertilized or activated parthogenically. You see the M2 oocyte lower than somatic cells. See the cumulus cell to the right? That's the typical somatic cell. The germ cell of the female, as I mentioned earlier, is consistently low. Same in human and mouse, monkey. But look what happens. As soon as we activate, there's a marked increase in telomere length. marked, even if we parthenogenically activate. Remember I said there's almost no telomerase activity? What the heck's going on? There's almost no measurable telomerase activity. And in fact, this led us to wonder whether there's an alternative method of telomere elongation going. It had been known for some time that there is an alternative pathway. It's called the alternative pathway, or alternative lengthening of telomeres, known as ALT. Look on the right there. Normal cells have a T-loop, that looped system. It protects the end, and then Telomerase would come along, unravel that, and use a reverse transcriptase to elongate it. But in the absence of telomerase activity in certain settings, cells destined to become sarcomas, embryos, that have very little telomerase activity, it's got two choices. Either can die, or it can develop the DNA recombination-based method called ALT. And in fact, we found ALT in all the strains. It wasn't just sort of one strain. All the ones we looked at, B63, CD1, C57 black. See, in the dark, fertilization, oocytes, zygote, two-cell. Parthogenic activation, even without sperm. You say, well, maybe the sperm is just adding into the embryo, and the aggregate of the two, when you measure the telomere length, it's just the sperm adding its longer telomeres. No, even with parthogenic activation, and even in telomerase null mice. So when we did this in null mice that had no telomerase activity, same thing. You could see wild type had the elongation. First generation telomerase null mice, elongation. Late generation, you can see they're starting to get pretty short. But even then, we found activation. They had evidence of telomere-sister chromatid exchange, which is how the elongation occurs. It's an invasion of one strand into the other in a DNA double-stranded break. We showed that the molecules that are involved in ALT, the DNA double-strand break repair machinery is present. RAD50, Bloom, Werner, these are proteins that both first unravel the DNA and then mediate double-strand break repair. The RAD50 MRA11 MRA11 is a complex we heard a little bit yesterday from Kutluk. But this was during the cleavage stage, and by the blastocyst stage, it is quieted down. And by then, we know from studies in human and mouse that by then the telomerase has kicked in. So you could see, in fact, there was a co-localization of the DNA double-strand break repair proteins in the telomeres. So that's kind of where we stood for a while. And in the spirit of this meeting, we want to also talk about what we're working on. What's new? What are we doing now? Well, it dawned on us that actually this is not a new mechanism of elongating telomeres early in development. And in fact, flies, Drosophila melanogaster, have been known for decades to elongate their telomeres, not through the action of telomerase, but through transposons. Transposons, as you recall, from basic biology, are these viruses that have been inserted into our genome over billions of years and are quieted by cytosine methylation in their promoter region. But during certain stages of development, specifically when the cytosine is demethylated as part of the wiping away, you can see activation by pol 2 of the RNA that reads off of the repetitive sequence. And then this moves into the cytoplasm, where it's actually reverse transcribed into a DNA, which is then reinserted. And usually, at least that retrotransposon or an associated retrotransposon also encodes reverse transcriptase, which is so essential. And in fact, there's extensive evidence that telomerase, the enzyme that's involved in elongation of telomeres during in stem cells and after the blast stage, and retrotransposons are very closely related structurally. You can see the orthology between line 1 and telomerase, and so at least it's been thought that it was pretty much only the fly that used retrotransposon activation to elongate telomeres. We argue that, in fact, that's what's happening, that during early development, we now know from many studies from many different groups that have shown that during early development following fertilization, even before transcriptional activation, there's a massive demethylation. There's extensive evidence that transcriptional activation is repressed, at least in yeast. It's increasingly understood on a molecular basis, but that there's a marked demethylation actively in the male pronucleus, passively in the female, and we argue that during this time there's activation of the line 1. This is a retrovirus that infected our ancestral genome billions of years ago. It's held in check through most development by methylation of these CpG islands in the 5' region. And this cytosine demethylation is stripped during early development to reset the epigenetic clock, which activates line 1. And in fact, line 1 is the only retrotransposon in humans that still has endonuclease activity necessary to form and also reverse transcriptase activity. So we wonder, this is what's going on? It's a very prevalent gene. It's 17% of our genome. In fact, if you include ALU and SINE, which are two other inactive retrotransposon, it comprises fully 40% of our genome. So we wonder whether this is involved. It's already been recently shown by a group at Hopkins and Carnegie that this is one of the drivers of fetal oocyte attrition, which is another phase of development when you get cytosine demethylation. So falling into this, it's been known for a long time that actually telomeres express an RNA. Recall that as part of retrotransposon activation, it starts with transcription of an RNA followed by reverse transcription into a cDNA, which then incorporates through double-strand break. And there's been a Tera molecule, but nobody really knew what it did. So we're looking to see whether this is the mediator of ALT. So this is very preliminary data where we looked in the blue, the normal oocyte, 2-cell, 4-cell, 8-cell elongation of telomeres, and you can see when we use 2 doses of AZT, a reverse transcriptase inhibitor, you can see we markedly reduce the elongation that's normally physiologically active. So in summary, and by the way, we've also done this in human. It looks very similar in humans, although those experiments, both sets of experiments are very active right now. So genomic instability is remarkably common in preimplantation embryos. It's mitotic as well as meiotic errors. It's mosaicism, partial mosaicism, copy number variants. There's no single explanation for the diversity of chaos that occurs. We believe that this origin is primarily nuclear, that telomeres are very essential for genomic instability, and their disruption can explain each of these manifestations, that a DNA recombination-based telomere elongation occurs physiologically, but it probably, part of the cost of doing business is extensive genomic instability. And so in this way, the old axiom that ontogeny really does recapitulate phylogeny. As we start in development, we take on these more primitive mechanisms. So I want to thank everybody knows Lynn. He did a beautiful job in his first talk of the day. Lynn and I have been working together for more than, 15 years? Is it more? It seems like just yesterday. A lot of the work done by Helen Wang, who came from Lynn's group and works with us, and a PhD student, Carrie, and our lab manager, Michelle. Thanks.
Thank you very much.